Welcome to Tomorrow Unveiled, where science doesn't just predict the future, it explains how you can live it better, right now. Here's something that should fundamentally change how you think about getting older. For decades, we've been told that cognitive decline is inevitable. Your brain peaks in your 30s, and it's all downhill from there. Your muscles weaken, your mind slows, it's just what happens. But what if that's not the whole story? What if there's a group of people, octogenarians with minds as sharp as people in their 40s, who figured out something the rest of us haven't? And what if their brains hold the key to understanding not just how to live longer, but how to stay capable, independent, and cognitively sharp for decades more? That's what we're exploring today. Because the emerging science of longevity isn't about extending life for its own sake. It's about extending the years when you can actually do the things that make life worth living. And it all comes down to resilience, the ability to maintain your function even as time passes. Let's start with the brain. There's a word that neuroscientists have been using more and more: superagers. These aren't comic book characters, they're real people. Individuals over 80 years old who have memories as sharp as people decades younger. Some of them can recall word lists, as well as people in their 40s and 50s. And their brains look different. Researchers at Northwestern University have been studying these superagers for over 25 years. They've recruited nearly 300 of them, autopsied almost 80 of their brains, and what they found is remarkable. Superagers don't just have sharp memories. Their brains have distinct anatomical signatures that protect them against the exact pathology that destroys everyone else. First, there's the plaque and tangle story. Most people develop amyloid plaques and tau tangles as they age. These are the hallmarks of Alzheimer's disease. They accumulate in the hippocampus, the memory center of the brain, and they cause the cognitive decline we all fear. But here's the wild part. Superagers either resist developing these tangles altogether, or they develop them and their brains somehow withstand the damage. Some superagers have very low densities of tau tangles. Others have moderate amounts, yet their memories remain exceptional. This suggests something profound, that there are at least two distinct pathways to maintaining memory in old age. You can either avoid developing the pathology in the first place, or you can develop it and be resilient to its effects. The researchers also found that superagers have larger entorhinal neurons, the cells in the brain's memory system. These aren't just slightly bigger. They're noticeably larger than neurons in younger people. What does that mean? It means the architecture of their neurons, the structural foundation, is sturdier, more robust, better able to form new connections and maintain functions even under stress. But here's what might be even more important. Superagers' brains show significantly less thinning of the outer cortex. That means less age-related shrinking, fewer lost neurons, and preservation of the white matter that protects signals throughout the brain. They also have less microglia, the immune cells that, when present in high levels, cause inflammation and neurodegeneration. The protective signature isn't about being disease-free, it's about being resilient, and that's a fundamentally different concept. Now, here's where it gets really interesting. Because researchers have discovered the mechanism that might explain how superagers maintain this resilience, and it's about something that scientists once thought was impossible: the brain growing new neurons in old age. For most of the 20th century, neuroscientists believed that the adult brain was fixed. You were born with all the neurons you'd ever have, and it was downhill from there. But in 1998, a landmark study challenged that assumption. Researchers found evidence that new neurons could be born in the hippocampus, even in adults. The field debated this for years. Some researchers claimed neurogenesis stopped after adolescence. Others found it continued throughout life. But a recent study published in Nature settles the question, at least for superagers. And the findings are striking. Researchers analyzed brain tissue from 5 different groups: healthy young adults, healthy older adults, superagers, people with early cognitive decline, and people with Alzheimer's disease. They used a technique called multi-omic single-cell sequencing, essentially scanning hundreds of thousands of individual brain cells to find developing neurons at different stages. Think of neurogenesis like a pipeline. You've got stem cells at the beginning, the raw material that could become neurons. Then neuroblasts, adolescent cells on their way to becoming neurons. Then immature neurons, young brain cells that aren't fully functional yet but are nearly there. Superagers had twice as many immature neurons as healthy older adults of similar age, twice as many. They had more newly developing neurons than young adults in their 30s and 40s. Meanwhile, people with early cognitive decline showed minimal neurogenesis, and people with Alzheimer's disease, they generated almost no new neurons at all. Dr. Tamar Giffen, one of the lead researchers, described it like this. Superagers are showing the preservation of immature neurons with heightened excitability. They are bright-eyed and bushy-tailed and ready to fire. That is a more youthful brain. But it's not just about the number of neurons. The hippocampus of superagers contains more robust support systems, specifically astrocytes and CA1 neurons that work together to create an environment that nurtures young brain cells. Young neurons are the most adaptable type of brain cell. They're plastic. They can grow, integrate, and wire themselves into the brain in ways that mature neurons can't. So superagers don't just have more new neurons. They have an enriched environment in their hippocampus that actively supports the growth and function of those neurons. It's like planting a young sapling in nutrient-rich soil versus rocky ground. And here's what connects to everything else we're going to discuss today. This capacity for neurogenesis appears to be something you can influence. It's not purely genetic. Lifestyle factors matter. Exercise matters. Cognitive engagement matters. The aging brain isn't fixed. It's plastic. It can change. You've probably heard that exercise is good for your brain. But what you might not know is that the same resistance exercise that builds muscle strength also appears to slow brain aging. And the mechanism is fascinating. When you do resistance training, lifting weights, doing bodyweight exercises, anything that challenges your muscles against resistance, you're not just stimulating muscle growth. You're triggering a cascade of molecular signals throughout your entire body. Your muscles produce compounds that circulate through your bloodstream and reach your brain, your bones, your cardiovascular system, your immune system. One of the most important compounds your muscles produce is klotho. It's a protein named after one of the fates from Greek mythology, the one who spins the thread of life. And that's fitting, because klotho appears to regulate lifespan itself. Klotho is one of the most fundamental longevity pathways we know about. It's a regulator of aging at a cellular level. When klotho levels are high, cells age more slowly. When they're low, aging accelerates. And here's the key. Muscle tissue is a major producer of klotho. When you exercise, particularly resistance exercise, your muscles pump klotho into your bloodstream. Studies in animals have shown that klotho enhances cognition, improves memory, and protects against neurodegeneration. In humans, klotho levels correlate directly with cognitive performance. People with higher klotho tend to have sharper minds. They score better on memory tests. They show less cognitive decline, but the muscle-brain connection goes even deeper. Resistance exercise also stimulates the growth of the hippocampus, the memory center. It increases blood flow to the brain. It reduces neuroinflammation, the chronic inflammation that damages neurons over time. It promotes the very neurogenesis we just discussed. So when you do resistance training and maintain muscle mass, you're not just maintaining strength and independence. You're activating one of the body's most fundamental longevity pathways. You're flooding your brain with protective compounds. You're stimulating new neuron growth. You're doing something that, based on the latest research, might be one of the most powerful interventions available for preserving cognitive function into old age. And here's something that might surprise you. This effect appears to be particularly strong in women. For women over 60, muscle strength is the single strongest predictor of longevity and health span, the years you live in good health. Let me ask you something. When was the last time a doctor measured your grip strength? If the answer is never, you're not alone. But that might be changing because researchers have discovered something unexpected. Your grip strength, the force with which you can squeeze your hand, might be one of the single best predictors of how long you'll live. This isn't folklore, it's not intuition. It's emerging from rigorous epidemiological research. Studies following thousands of people over decades have found that grip strength correlates with mortality from all causes. It predicts cardiovascular disease risk. It predicts cognitive decline. It predicts frailty, disability, and loss of independence. In fact, grip strength is such a robust predictor that some researchers are proposing it become a standard vital sign, right up there with blood pressure and heart rate. When you visit your doctor, they should be measuring your grip strength. Why? Because grip strength is a proxy for overall muscle mass and muscle quality. It reflects your ability to maintain the physical capacity to move through the world. And as we're learning, that capacity is intimately connected to every system in your body: your brain, your heart, your bones, your immune system. Here's the thing that makes this so important. Muscle loss, sarcopenia, is one of the defining characteristics of aging. Starting around age 30, most people begin losing muscle mass at a rate of about 3 to 5% per decade. By the time you're 70, you might have lost 30 to 40% of the muscle mass you had at 30. But this isn't inevitable. You can prevent it. You can reverse it. And the tool is simple: resistance exercise. When you maintain muscle strength, you maintain the ability to move through the world. You maintain independence. You maintain the ability to recover from illness or injury. You maintain the biological signaling that keeps your brain young. Your muscles aren't just for moving things around. They're endocrine organs. They produce hormones and proteins that regulate aging throughout your entire body. And grip strength is just one measurable marker of this broader capacity. It's the tip of the iceberg. Your doctor probably checks blood pressure, cholesterol, diabetes markers. These are disease markers, indicators that something is wrong. But what if we're measuring the wrong things? A new paradigm is emerging in medicine. Instead of asking, "Are you diseased?" we're starting to ask, "Are you functional? Can you move? Can you think? Can you maintain independence?" Functional aging metrics are measurements of what your body can actually do. Grip strength is one. Walking speed is another. People who walk slowly have higher mortality risk, even if everything else looks normal on their blood work. Ability to rise from a seated position without using your hands. Balance, ability to climb stairs. These aren't arbitrary tests. They're measures of your capacity to live independently and maintain quality of life. But there's something even more precise emerging: biological age diagnostics. These are tests that measure your aging at a molecular level, not how old you are in years, but how old your cells actually are. Biological age can be estimated through various methods. Some look at telomere length, the protective caps on your chromosomes that shorten with each cell division. Others measure epigenetic markers, chemical tags on your DNA that change with age. The most sophisticated tests use combinations of blood biomarkers to create an overall picture of your cellular age. And here's what's remarkable: your biological age and your chronological age don't always match. Some 60-year-olds have the cellular profile of 40-year-olds. Others have the profile of 80-year-olds. The difference? Lifestyle factors: exercise, diet, sleep, stress management, social connection. Your muscles, your brain, your cardiovascular system, they all have a biological age, and they're not fixed. They can get younger with the right interventions. This is why functional aging metrics and biological age diagnostics are becoming increasingly important. They're not just interesting data points, they're actionable information. They tell you whether your interventions are actually working. They tell you whether you're getting younger or older at a cellular level. And they're reframing the entire conversation around longevity. It's not about living to 100, it's about maintaining the functional capacity to enjoy those years. It's about keeping your biological age aligned with, or even younger than, your chronological age. Hey, real quick, if you're enjoying this, do me a favor and hit that subscribe button and turn on notifications so you don't miss the next one. We're diving into some seriously groundbreaking territory here. What if frailty, the condition that seems irreversible, actually isn't? What if you could reverse it? Frailty is a specific clinical condition. It's not just being old and weak. It's a syndrome characterized by loss of muscle, loss of strength, loss of endurance, reduced activity, and increased vulnerability to stressors. A frail person might fall and break a hip. They might develop an infection that cascades into serious illness. They might lose the ability to live independently. For decades, frailty seemed like a one-way street. Once you crossed that threshold, the assumption was that decline would continue. But recent research is challenging that assumption. One of the most exciting developments is the use of mesenchymal stem cell therapy. Researchers at Longeveron, a regenerative medicine company, conducted a clinical trial using stem cells from young, healthy donors to treat age-related frailty. The results were striking. In a Phase 2b trial, patients with age-related frailty received an infusion of allogeneic mesenchymal stem cells, cells derived from young donors that have the capacity to reduce inflammation, promote tissue repair, and support cellular regeneration. The results showed significant improvements in functional capacity. Patients could walk farther. They had better balance. They showed improved strength and endurance. One particularly compelling marker? The 6-minute walk test. This is a standard measure of functional capacity. How far can you walk in 6 minutes? Patients in the stem cell treatment group improved by an average of 63.4 meters. That might not sound like much, but in functional terms, it's substantial. It's the difference between being able to walk to the mailbox and not being able to. It's the difference between independence and dependence. And here's what's remarkable. The improvements persisted. Patients didn't just improve temporarily. The benefits lasted. The mechanism appears to involve reducing systemic inflammation, the chronic, low-grade inflammation that characterizes aging and drives frailty. As we age, our immune system becomes increasingly dysregulated. It produces chronic inflammatory signals that damage tissues throughout the body. Stem cells appear to reset this inflammatory state, allowing tissues to repair and regenerate. Longeveron is also testing stem cell therapy for Alzheimer's disease using a product called laromestrocel. Early results are promising. The company has published data showing improvements in cognitive function and quality of life in Alzheimer's patients. Now, stem cell therapy isn't a magic bullet. It's not a cure, but it represents something profound. The recognition that aging is a reversible process, that frailty isn't destiny, that the body has remarkable capacity for repair and regeneration if we give it the right tools. And this ties directly back to everything we've been discussing. Stem cells work partly by promoting neurogenesis, by reducing inflammation, by restoring the cellular environment that supports healthy aging. They're not replacing the need for exercise and good lifestyle choices. They're amplifying the body's natural capacity for resilience. So far, we've been talking about things you can do. Exercise, maintain muscle, engage your brain, but there's also a genetic component to longevity. And understanding that genetic component is opening up new therapeutic possibilities. Researchers have identified genetic signatures associated with exceptional longevity, the genes that appear in people who live to 100 and beyond. These genes are involved in several key pathways: inflammation regulation, cellular stress response, DNA repair, and metabolic health. One of the most important is the Klotho pathway we mentioned earlier. People with certain variants of the Klotho gene tend to live longer and maintain better cognitive function. But here's the key insight. Klotho expression isn't fixed. It's regulated by lifestyle factors. Exercise increases Klotho production. So does caloric restriction and intermittent fasting. So does cognitive engagement. Another important pathway involves NAD+, a molecule that's essential for cellular energy production and DNA repair. NAD+ levels decline with age, and this decline is associated with aging and age-related diseases. But NAD+ production can be stimulated through exercise, certain supplements, and caloric restriction. There's also the SIRT pathway. Sirtuins are proteins that regulate cellular stress response and longevity. They're activated by caloric restriction and exercise. They protect cells from damage and promote repair. What's emerging is a picture of longevity as a network of interconnected pathways. You don't have to optimize all of them perfectly, but if you're activating multiple pathways through exercise, good nutrition, cognitive engagement, and social connection, you're amplifying your body's natural resilience to aging. And here's what's fascinating. Many of the interventions that activate these longevity pathways, resistance exercise, cognitive challenge, social engagement, are things you can start doing today. You don't need to wait for new drugs or therapies. You can begin activating these pathways right now. The genetic component isn't destiny. It's more like a blueprint. Your genes load the gun, but your lifestyle pulls the trigger, or doesn't. Here's what connects all of this research, a consistent theme that's emerging across neuroscience, gerontology, and molecular biology. It's not about being perfect. SuperAgers don't all eat perfectly. Some have heart disease. Some don't exercise as much as they should. Some have tau tangles and amyloid plaques in their brains, but their brains are resilient to the damage. Frail older adults can't be fixed by making them healthier. They can be fixed by restoring their capacity to recover from stress. That's what stem cells appear to do. Muscle isn't just about strength. It's about the signaling that keeps your entire body young. Clotho, NAD+, sirtuins, growth factors. All of it flows from maintaining muscle mass. The brain isn't fixed. It's plastic. It can grow new neurons at 80, 90, even 100, but only if the conditions are right. Only if you're challenging it. Only if you're moving your body. Only if you're maintaining the biological signals that support neurogenesis. And here's the profound insight. Resilience isn't about preventing all damage. It's about maintaining the capacity to recover, repair, and adapt. It's about keeping your systems flexible and responsive. This is why functional aging metrics matter more than just chronological age. This is why grip strength matters. This is why biological age testing is becoming important. They're measuring resilience. The emerging picture of longevity is fundamentally different from what we thought even 10 years ago. It's not about finding the one magic intervention. It's about understanding that your body is a network of interconnected systems, all of which can be optimized. It's about recognizing that aging is plastic, changeable, not fixed. And it's about understanding that the goal isn't just to live longer, it's to stay capable, independent, and cognitively sharp for as long as possible. It's to compress morbidity, to push all the years of decline and disability toward the very end of life rather than spreading them across decades. You've got SuperAgers in their 90s with memories as sharp as people in their 40s. You've got people reversing frailty with stem cell therapy. You've got evidence that your brain can grow new neurons at any age. You've got proof that your muscles regulate aging throughout your entire body through the proteins they produce. This isn't science fiction. This is what the research shows. Right now. Oh, and if you're watching on your phone, there's a hype button right below the video. Smashing that really helps channels like ours get discovered. So what does this actually mean for you? How do you translate all of this research into action? First, prioritize resistance exercise. Not cardio, not just walking. Actual resistance training, weights, resistance bands, body weight exercises. This is the intervention that appears most consistently across all of this research. It stimulates neurogenesis. It maintains muscle mass and strength. It produces clotho. It activates longevity pathways. If you do one thing, do this. Second, measure what matters. Get your grip strength tested. If you can, get your biological age assessed. These aren't just vanity metrics, they're actionable data. They tell you whether your interventions are working. They motivate you to keep going. Third, engage your brain. Learn something new. Read. Have complex conversations. Solve problems. The brain that's challenged is the brain that maintains neurogenesis. SuperAgers tend to be cognitively engaged throughout their lives. They don't retire mentally. Fourth, maintain social connection. SuperAgers are social. They have strong relationships. They're engaged with their communities. Loneliness is a risk factor for cognitive decline and premature death. Connection is protective. Fifth, consider your diet and sleep. We didn't dive deep into these in this episode, but they matter. They affect NAD+ levels. They affect inflammation. They affect your body's capacity for repair and regeneration. And sixth, understand that this isn't all or nothing. You don't have to be perfect. You don't have to optimize every pathway. But if you're activating multiple pathways through exercise, cognitive engagement, social connection, and good nutrition, You're doing something profound. You're building resilience. You're keeping your biological age younger than your chronological age. You're maintaining the capacity to live independently and enjoy life. The research shows that aging isn't destiny. Your brain can grow new neurons. Your muscles can stay strong. Your mind can stay sharp. Frailty can be reversed. Your biological age can get younger, but only if you do the work. The question isn't whether it's possible, it's whether you're going to do it.
Let the future be your guide with all the